Hey, Alex, great to have you on the podcast. How's it going?
Oh, thank you. Thank you for having me here. It's going great, although it's too dark in Finland right now. But anyway, I'm really happy to be here and participate in the first of its kind podcast devoted to such a such an important and I will say like underestimated or overlooked topic, right? As battling then defeating aging eventually. So happy to be here.
Great. And I think a really good place to start would be for you to tell us a little bit about your journey. How did you find your way into this field of aging/longevity?
Yeah, so it is rooted into like my childhood, or when I was like a child, I thought like, okay, I don't want to be old. Just why people become old and die eventually. I've experienced several deaths, and of course, like many of us do, from relatives. And my granny died from cancer, for instance, and I witnessed it. And of course, it seemed to me like totally unnatural and unfair. Why our beloved ones leave us? But at that time, I didn't pay too much attention to the aging research field, and I was not trying to do something by myself, right? Because I thought like, okay, the science advances like at in incredibly fast pace, right? In 5 or maximum 10 years, the therapies that can slow down or even reverse aging will be there and at our disposal. We can probably use it, and then it will become cheaper and cheaper and more people can afford. But as time passed, I started to notice that the progress in some fields is, let's say, incredibly fast, right? But progress in a very certain fields that are the most interesting to me, like medicine, anti-aging biology, whatnot, right? Kind of related things. It's the pace is extremely slow. And then I started to think, wow, so what's wrong with that? So to me, it seemed like pretty simple problem, right? Well, we age. Some other things also do age as well. So, but people somehow can repair, for instance, machines. So why we can't repair our bodies, right? And then I started to think about it. Unfortunately, or fortunately, I don't know, like what is the time will tell. But I wanted to become a biologist or medical doctor or something like that. But I chose another path. So I became a computer scientist. So I have a master's degree in computer science, and I worked on AI. Yeah, so I was developing a chess program that learns how to play chess, but by playing with itself. Yeah, so it was an incredible journey. And later I got like very positive vibes when I learned that DeepMind did kind of the same, but on of course much larger scale than me. And they succeeded greatly by defeating like Go champion, like basically anyone at Go and chess also. So computers totally dominate in these fields. So, but I always was into biology. Still, I was interested, and then I was positively surprised when I discovered that there is a field of bioinformatics, so which kind of brings two favorite worlds of mine together, right? So biology and computer science, machine learning, statistics, AI, everything is kind of connected. And yeah, I started to learn bioinformatics, participate in competitions, like various competitions, online competitions. There was like Rosalind project or something like that, where people solved like different programming tasks, from the bioinformatics area. And then I found a job in research institute of antimicrobial chemotherapy. So that was a little bit different from the field of aging, but I learned a lot of microbiology there, molecular biology, and learned how laboratories operate, various techniques, what is sequencing, what is mass spectrometry, and so on and so forth. And then I was happy to meet online. I was a very young and dare man. I contacted one of my teachers, so to speak, Alexei Moskalev. He's a professor and one of the most famous researchers in the field of aging in Russia. So nowadays, and back then also, like some time ago. And basically since then, we started to collaborate. That was like, I don't know, like 2011 or something like that. So more than 10 years ago. And yeah, since that time, we started to collaborate. And initially my focus was on drugs that extend lifespan. They are also known as geroprotectors. So I hoped, and I actually believed at that time, that by combining several drugs, we can get like not even additive but multiplicative effects, right? So when combination has much powerful synergy between components that can extend lifespan significantly. But then I was kind of disappointed in that idea because like evidence tells the opposite. So it is easy to find a combination of drugs that worsens the effect of the most powerful drug, rather than find a combination of drugs that mutually increases the efficiency of every drug alone. But still, the idea is not totally wrong, and there are combinations that are very promising. For instance, rapamycin and acarbose. This is like, in my opinion, a breakthrough in this field. But anyway, so I realized that okay, we are not gonna achieve like radical life extension in humans by looking into small molecules, into drugs, and to find a real breakthrough, we need to understand the mechanisms that, well, basically drive in aging. And then I started to dive deeper in the molecular pathways associated with aging. And I discovered one very nasty thing, that that all these pathways, they are kind of looped. There is a circular dependency. If you try to find what causes what, you basically go in circles, right? So, for instance, inflammation causes, I don't know, like muscle, like stem cell depletion or whatever. But what causes inflammation? And then we return back after several steps. It turns out that inflammation causes inflammation, or there were some attempts to, like maybe DNA damage is the key driver of everything, right? Because there are evidence that DNA damage can cause multiple downstream effects, right, that are observed during aging. But unfortunately, there are quite some studies that introduce like more mutations per unit of time in various animals, including mice, and it doesn't seem to support the hypothesis that DNA damage is the key driver of aging. Of course, it plays a role, but probably not a decisive role, and can't be named as a cause of aging. And of course, there might be more than one cause of aging, right? And they can play at a different times. But let's talk about it maybe later today. And then, so I realized that there should be some starting point, because otherwise we can't, remove this loops, this circular dependencies. So something should just be a launcher for that avalanche of aging, right? And then I don't remember how exactly, but then I got back to this idea of something that accumulates damage just by the design. So our bodies mostly recycle all the damage, because we have DNA damage repair mechanism, we have lysosomes, we have mitophagy, autophagy, we have various detox enzymes. We have protein folding different structures that help the protein folding. But we still have some damage that is not repaired in principle. So, well, I realized, okay, if this damage is not repaired, or like virtually if it accumulates, it might ignite this fire of aging, because, well, that's the logical candidate. If there is a flaw in design of the system, that it might be a driver of cascading failure of the whole organism.
You mentioned like aging is these sort of loops, or I would think of it maybe more as like a network of all these different processes that all affect each other. And right, so if you accumulate damage or deviations from homeostasis on one of those processes, it affects all of the other processes, and that actually kind of lines up well with the way you see sort of mortality curves have this sort of upward sort of trajectory, right? It's an exponential process, because as things start to go wrong faster because they're affecting each other. And geroscience traditionally has been, people like to look for linear chains of cause and effect, because if there are linear chains of cause and effect, that's very convenient, because then you can intervene. But if the true nature of the problem is that there is no linearity, there's no reductionist way of thinking about it, that's a problem, because then you actually have to fix everything. You can't just fix one thing and hope for that it'll solve all of the other problems. And so it seems like you kind of both agree with the fact that there is like this sort of network, but you also still think that there may be, I guess, more important nodes in the network, or at least you're hoping to. And I think we're going to get into more detail there, but I guess I want to challenge you on like, is there actually a more important node of the network? Like, and how do we know that?
It's a very good point, and I'm not saying that there is like, if you remove some damage that initiated the cascade of failures, then the system will return back into useful state. But what I was trying to find is what is likely to cause this chain of events that lead eventually to the death of the organism. So the causal node, right? So because it's like an avalanche, right? I've mentioned this before, right? So a small deviation from the equilibrium state can cause it to fall, right? But after it falls, so if you remove something that initiated the avalanche, it won't help, right? But luckily, there are signs that at least some of the critical things can be restored or mitigated if we fix like damage to long-lived macromolecules.
Yeah. The differentiation between things where we know there's inbuilt repair capacity, even if that repair capacity declines, at least it does exist in principle. But then there is a category of things that it seems like there just is actually not inbuilt repair capacity for. And I think that's like something that you've highlighted in your research.
Yes.
So like what do you think are those things that we just simply don't have an endogenous ability to repair?
Well, that's a very good question. So we know for sure some of these molecules, but of course there is also a huge territory of unknown. So I will highlight only what we currently know, right? And most people know my work as like a matrix or ECM hypothesis of aging, and this is because majority of these long-lived proteins are in the ECM. And there is a good reason of why this is happening, right? So why these proteins are long-lived and why they are not repaired, because most of these reparation enzymes, they are in the cells and they maintain this cell integrity and the cell functioning. But outside of the cells, the concentration of enzymes is very low. If non-zero, right? So, and that's why these molecules get damaged, right? So, for instance, collagen has a half-life dependent on the tissue, but we can say like roughly 10 to 15 years, right? Elastin, for one, another very important protein, is like half-life of more than 100 years. So we don't know exactly, but presumably it basically is deposited only once per lifetime and then is just subject to degradation, like continuous degradation. There is another very important and yet very unexplored area of nuclear pore complexes. Nuclear pore complexes in non-dividing cells are very extremely long-lived. At least in rodents, it was shown that they live for the entirety of the organism lifespan. And since we are kind of don't know exactly what causes Alzheimer and other dementias and cognitive problems, that might be also a good candidate, right? Good target, good mechanism, because when cells divide, they reassemble the nucleus and also like create this nuclear pore complexes de novo, like from scratch. But in non-dividing cells, these proteins remain basically intact.
Yeah, and I think that's a very important distinction, the non-dividing versus dividing cells,
Yes.
which is also related to like multicellular versus, single cellular life, right? Like multicellular life is more recent evolutionary. And so it makes sense that cells have had time to evolve mechanisms to keep their internals, functioning. But before we were multicellular, cells would just always divide, and so they could always just dilute certain types of damage. And then we became multicellular, and we started having somatic non-dividing cells, and we had to evolve ways to keep them alive. But perhaps that's just an incomplete process, right? Natural selection has not yet sort of built all of the things necessary there. I guess, yeah, what do you think of this in terms of the evolutionary nature? Because again, like the extracellular matrix is also something that's relatively recent by evolutionary standards, you could say. And so that might be why we don't have mechanisms to completely repair it. Yeah. Do you think about this in an evolutionary context, or do you think that's just more too speculative?
Yeah, you caught me off guard. So I actually think little in terms of like evolutionary. So I know a lot of people, not a lot of, but I know some very smart researchers who try to look at aging from the theoretical standpoint. So from evolutionary view, from understands why aging is needed. Is it programmable, or is it like stochastic damage? I'm more on an applied side of things, so I try to solve the problem. So that's my motivation.
I mean, I agree with you with that, and like ultimately, I mean, how we got into this situation may or may not be relevant unless it implies a possible solution.
Yes, exactly. But yeah, so in terms of evolution, I thought only about like whether the agent is programmable or not. And because it is actually quite an important thing, because what programmable hypothesis of aging tells us is that, okay, we have a perfect capacity to repair tissues, but at some point in time, the kill switch is turned on, and there's some mechanism that starts the killing cascade, right? So that the repair of tissues switches off, and all bad things happen to the organism. And this is very important because it offers a nice solution. So if we learn how to prevent this, how to switch off this killer program, or how to trick it in some way, then we are basically saved. And this is a very attractive idea. I understand people who try to find this program of aging, but since I've already mentioned that okay, we have already parts of the body that are not repaired, essentially, this is already a good explanation. So why aging is happening, and I think this is a little bit of a redundant hypothesis, not parsimonious enough to be taken seriously. However, I would like to emphasize that we are not in the situation to refute everything but one hypothesis. So since our goal is to eventually, extend our life radically, so to speak, we don't have this luxury to, refute points of view that we don't like, that do not fit into our point of view.
Yeah, although I think part of the exercise of this podcast series is what we want to try to get at is, for any given approach, what are the underlying assumptions and how could you test those assumptions, right? So like the idea of programmed aging has been around for a long time, and people have tested it in various ways, and I think for the most part it's failed to hold up, although it's hard to entirely eliminate it in the sort of cellular reprogramming field right now.
Very difficult.
I think the cellular reprogramming field, I think, maybe makes the strongest case for that. But maybe we could get to that later if you want. But I think there's a lot of sort of the issues you highlight with the extracellular matrix are, I guess, probably one of the biggest potential flaws in the people who are the strong advocates for cellular reprogramming being a complete solution. I don't know if you want to comment on that.
Yeah, definitely. First of all, it's a little bit, I don't know. I feel a little bit, I disagree with people who claim that cellular reprogramming is about programmed aging theory, right? Because cellular reprogramming is, I don't know, it may very well be that it has downsides, and we know about it. So recent paper claims that, for instance, if we induce cellular reprogramming everywhere, just animals die, right? So that's why it might be a very strong reparation mechanism that is put on hold because of that reason, right? Because if it's on a multicellular, multi-tissue organism, if it's turned on, the organism dies. But for a single cell, it might be just good enough to clear all the damage and start everything from scratch. And this is supported by like a plethora of evidence, because we now know for sure that at least some somatic cells, not even germ cells, right? Not even germline, but somatic cells can outlive an organism by a factor of at least 3. So 3 times longer than the maximum lifespan. So that was shown on hematopoietic stem cells in mice, on T lymphocytes recently in mice as well. That was also shown, though to a lesser degree, for neurons that can live like 30 to 40% longer than the maximum longevity of an animal.
Yeah, this is actually really interesting work that I think probably not a lot of people are aware of. And this might be an opportunity if you want to share some of your slides and actually explain this research to people. I think it would be something that a lot of people haven't heard about and would benefit from seeing.
Yeah.
So the people listening at home will try to explain a little bit.
Yeah, there is not much on the screen. This is a copy of an article from PubMed. This is quite an old article. It's from 1979. So people back then already were aware of that. So what did the researchers do? If I remember correctly, because I haven't read this paper for a while, they have mice that had killed bone marrow, right? And then transplanted bone marrow, essentially hematopoietic stem cells from another animal. And this bone marrow lived in this animal for the entirety of its life. And then it was, in a similar fashion, it was serially transplanted to another young mouse, right? And this was done 3 times, if I remember correctly. And we know that for sure because mice were to whom this bone marrow was transplanted, they were not able to produce blood cells. But when the old bone marrow was transplanted, they began to produce blood cells normally. And the interesting fact that in this study, not the time itself was a critical factor determining the lifespan of these stem cells, but the number of transplantations. And the researchers concluded that this transplantation procedure was somewhat harmful to the cells. And theoretically, the lifespan of individual cell in an ideal environment could be much, much longer than we can imagine. So it is already was shown to be 3,000 days, while typical mouse lifespan is around 800 days, like median longevity. So that's incredible in my opinion. But then you can say, okay, that's a very old paper, right? People may do whatever mistakes, conditions, whatever. So people do not trust usually old science, right? But anyway, very recent paper, January 2023, roughly the same procedure repeated with T lymphocytes. It's not even stem cells. These are somatic cells which, execute immune function, right? And basically the same, numerous transplantations, also several fold longer than lifespan of the organism. And interestingly enough, this immune cells, transplanted immune T cells, were still capable of executing their function, right? So they were functional T cells capable of killing infectious agents. And this is a study about neurons. And this is also a very, very, very interesting study where scientists took neurons or neuron progenitor cells from mice and implanted them into rat brains. And this neural progenitor cells, they successfully incorporated in rat brain, which is by itself a very remarkable thing, right? So neurons from one organism work in the brains of another organism. And here I say hi to my friend Jean Hebert, who is doing kind of very similar work. Yeah, greetings to you from Finland. And then so these neurons lived for the entirety of the lifespan of a rat. And rats live 30 to 40% longer than mice. And that disproves the initial claim or initial belief that was back in the days that, okay, cells in this particular region of the brain start to die, and this is what causes the cognitive decline and aging of the brain. That just cell death is the main contributor. No, it's not the case. So cells do not die. And also, for instance, recent research, a very cool study that gives us hope, that showed that, for instance, sarcopenia is the muscle loss of muscle mass. It is, one hypothesis was that it is caused not by loss of so the muscle cell loss is secondary. The first is loss of motor neurons, which innervate the muscles, right? And if they die, muscles do not get signals from the brain, and also shrink in size and die eventually, because if you don't use it, you lose it, right? But very recent study, also from 2023, discovered that motor neurons do not die. So the quantity of motor neurons remains the same. It's just the number of excitatory synapses that decreases with aging. And it means that they send less excitatory signals, right? Less positive motion signals, and less stimulation, and less usage of muscle tissue, and decline. But the neurons themselves do not die. It's just something changes in the way inside the cell, and this leads in turn to this weird communication problems between different types of cells.
Yeah, I agree. It's important because if you're going to be trying to do cell-based replacement therapies, but loss of cells is not the
It's not
actual
gonna
problem,
help.
it's not actually going to help,
Yes.
is the implication.
Yes, this is the problem of that cells somehow adapt to something, right? And adapt in a weird way that breaks alters the function that they normally do, right? And this adaptation, in my opinion, is mediated by the environment. So cells adapt to the environment and then lose their ability to function properly. So, and this environment is mostly ECM, and ECM has a lot of long-lived molecules that are cross-linked, or there are adducts, and there are multitude of ways how it can influence the cellular signaling, starting from mechanosensing to like receptors on the surface of the cells and whatnot.
So I want to challenge a little bit on this, and in that, so these experiments where you're transferring cells, from one animal to another, have they accurately quantified what percentage of those cells actually are able to continue? Because these are dividing cell populations often, and it may simply be the case that cell loss is still an issue because there's a selection happening.
Of course. And this is, I don't think that the study from 1979 has done this clonal analysis, right? I'm not sure. I need to reread the paper from 2023 about the T cells. Not sure, but you mentioned.
Yeah, I think an experiment where they actually did clonal analysis to show truly what portion of these cells and are there clonal populations that do die off versus not would be very valuable information.
So if some clones survive and basically during the transfer and normally execute their functions, I think it's not a problem. The problem is that when the clones that are selected, they become less functional, so to speak. And this is exactly what happens during aging of our bone marrow, or like hematopoietic stem cells. So normally, cells in young organisms, they are lymphoid biased. In the middle of age, there is like an equilibrium between lymphoid and myeloid branches. And for the listeners who are not very deep into biology, I can say a couple of words what lymphoid and myeloid branches are. So lymphoid cells are components of an adaptive immunity. So the immunity that is against viruses and sometimes cancer, or other malignant tumors and whatnot. While myeloid cells, they are part of non-specific immunity, pro-inflammatory cells, and we know that inflammation is a companion of aging, right? And then they also execute antibacterial immunity and like multiple other things, but tissue repair, whatnot. But the main feature is that they are a part of non-adaptive immunity, and many of them are pro-inflammatory. And this is bad because the balance shifted towards myeloid cells is creating this non-healthy pro-atherosclerotic, pro-atherogenic environment, and basically we are more susceptible to viral diseases, which is a case of COVID showed to us, right? So that the highest mortality rate is in the group of people older than a certain age. Well, in fact, it basically continuously grow with time, with age. But yeah, so the highest risk is in the elderly population. And then basically we know that clonal hematopoiesis happens. So some clones during aging get an advantage. So they basically outcompete other cells. And unfortunately, these clones are myeloid biased mostly. So they produce this pro-inflammatory cells of non-adaptive immunity. And interestingly enough, this grows exponentially. So here is the nonlinearity that we were discussing previously. So, and the fraction of this clonal hematopoiesis is extreme. It's like more than 50% of people have clonal hematopoiesis after the age of 85. So it will eventually, everyone will have this clonal hematopoiesis. And this is something that limits
This is very
this is something that limits our lifespan for sure. I'm absolutely sure.
I want to jump in here because when people see the mortality curve and the risk curve for age-related diseases, it always has this exponential sort of like function with age, right? And this is well known, like the Gompertz law of mortality and so forth. And one of the things that I remember Peter Fedintsev saying, maybe I don't quote me on this, but one of the things he was talking about in his journey to try and figure out like what is causing this aging, how do we solve this problem? They were trying to find something that physically was also increasing exponentially as you age. And if you look mostly in the body, I think there's not that many examples of something that actually grows exponentially with age that you can actually like physically measure, like a quantity that is actually changing, not like a risk sort of measure. So this is really interesting. I think this is one of the few things that I've seen where it's actually increasing exponentially, not like linearly.
Yeah, I actually was also looking at some point in time. I was also looking at things that grow exponentially. It's kind of an attractive idea, right? And the only thing that I was able to discover were the number of senescent cells. And this is like a pattern. So here are also cells. And this is not surprising because cells divide. And division is like an exponential process. I think there is some connection between this fact. So, but anyway, I think a wild idea like came to my mind, right? So what may cause this exponential? So the environmental aging, so like extracellular matrix or some other components that have long-lived like molecules, polymers, whatnot, very slowly it creates an environment which gives a selective and like a competitive advantage to certain cells that may or may not execute their functions properly. And it happens not only with hematopoietic cells, but like in many places as well. For instance, in the colon, or in the epithelium of lungs, or in stomach, where cell division rate is high. And basically that may, and because of the these different clones, they may have different epigenetic marks, right? The composition of cells changes because of the clonal selection. And so these certain epigenetic marks, these clones might not be even genetic, but also epigenetic. So some cells might have like epigenetic switches in certain places. They might, and these epigenetic mutations can give them advantage in a certain environment and certain state of the extracellular matrix. And this is what epigenetic clock may capture. So this was one of my recent ideas.
Yeah, I think just for the, I guess, more casual listeners, Andrew Steele's book Ageless does talk about clonal amplification and what he calls clonal mosaicism as what accumulates with age, and he describes it in a fair amount of detail. So I recommend that book for somebody that needs some background to this area. And like you say, it's not just the hematopoietic cells. This is happening in other cell populations as well, particularly the ones that divide, including your skin, endothelium, the gut, all over the place. And I think if it's being driven by epigenetic changes, that's, I guess, maybe more hopeful because that's something that you could potentially reverse. If it's being driven to the extent that it's being driven by nuclear mutations, that's a harder problem. I think that's something that we'll be interviewing other people on who specialize in studying that. But is there, I guess, in your own research, I mean, it sounds like obviously the extracellular matrix is one of those potential drivers. What about nuclear DNA mutations? Is that something that you have thought about and investigated as a potential other driver that would be difficult to deal with?
Yeah, so as I mentioned, I studied DNA mutations back then, but there are lots of evidence that it is unlikely to cause aging per se, or like in several organisms, right? But on the other hand, I think even if this clonal hematopoiesis or clonal selection happens on a DNA level because of the DNA mutations, it is also not a game stopper, so to speak. And we probably don't need to fix even DNA. We just need to create conditions that favor cells that still execute normal functioning. So we need to remove this selective pressure, or even introduce a new selective pressure that favors good cells, so to speak. And then it will naturally, this clonal selection will play on our side. So I don't think it's like a huge problem. So we can turn it to play on our side. So we don't need to fix like every DNA mutation. We just need to allow natural selection do the job.
Yeah, mitochondria are sort of maybe a potential case here, right? Because there's been long hypothesized that there's a selective advantage to mitochondria that have certain mutations. And I think there's some evidence behind that. And there's our proposals for how you could change that selective bias to favor the functioning ones. Is that an area that you've thought about?
Yep. Yeah, this is something that we are actively. So currently, I'm a chief science officer at Radical Life Extension Group. This group is mostly focused on, biohacker means of working, right? So we're trying to, research cutting edge, interventions for extended lifespan. And also I'm a research director at Elastin Biosciences. This is a startup aimed to restore elastin. But yeah, most of the time I dedicate to Radical Life Extension Group, and clonal hematopoiesis is one of the things that we are currently working on, like trying to combat it. And the thing is that the most probable candidate for this selective pressure is inflammation. But this inflammation is very localized. So it's not, you probably cannot detect it even by high sensitive CRP or some other blood biomarkers of inflammation. So this is probably a localized inflammation in the environment, in the niche of hematopoietic stem cells. And there are evidence that some like modern drugs, for instance, like this MABs, like canakinumab or anakinra, so interleukin 1b blockers and some other, they actually reverse some effects of aging of stem cells. And this is like extremely interesting. So if we can block it in vivo and show the results, like and see at the composition of clones in stem cells, and that this gives us hope that we can see something, so some changes, favorable shifts in the clonal composition of the stem cell pool. But first we need to learn how to measure this stem cell composition. So taking bone marrow every time to do an analysis is an infeasible procedure, right? So that's why we are working on the extraction of cells from peripheral blood and basically working with this population and trying to understand what markers can serve as a proxy. So how can we, using cytometry and other stuff, how can we, create a relevant picture of what's happening in the bone marrow?
Great. So I want to step back a little bit, because the whole point of this series that we're doing is to try to build a actual roadmap, because we think that having a credible plan for how to address aging through multiple strategies would actually make it easier for both more people to enter the field and do the work, but also for more investment to flow into the field.
Absolutely.
And so we want to actually build budgets and timelines and say, hey, these are the things we could try. This is how long it would take. This is how much it would cost. And your specialty here is dealing with the clonal populations, the problematic clonal population growth, and the extracellular matrix. So I guess my question is, well, first, I think it might be worth spending a little bit more time just explaining the things that do happen to the extracellular matrix and how sort of, the diversity of changes that happen and how the scale of the problem. But then have you actually thought of like a roadmap to what it would take? Like, what are the tools? What are the experiments that need to be done? What are the tests that need to be done in order to actually try to solve this problem of extracellular matrix? Or what are the technologies that need to be developed? Yeah. Do you have a roadmap in your head or that's written down?
Yeah.
So those are my two questions.
It's not written down. It's in my head, unfortunately, but it's a good reminder for me to put everything on paper. But before that, there were no place to display such a roadmap. But I think it's a good starting point. So if we would have like a place where we can share different views and concepts and roadmaps, I will definitely prepare something more better detailed and yeah.
No, I mean we're very much down to actually give you support in making that happen and helping build that roadmap. That's actually going to be one of the primary missions of the LDF going forward. So for now, maybe though you could at least start by like I said, outline the scope of the problem. How hard is this problem really with the extracellular matrix? And I think you have some good slides on that. And then just give us an outline of what do you think it would take? Like, what are the technologies that we would need? And we can figure out the budget and the time estimates later, but maybe just outline the scope of the problem first.
Yeah, it's a very good point. So, and then we start from collagen insolubility problem. And this is, I think, real baddie today. So so first of all, we know to some extent that collagen affects the stem cell division, or this is stem cells, and basically cells on young ECM obtained from young cells divide better than cells grown on ECM obtained from old cells. There are like multiple studies that show exactly this thing. But what actually happens with collagen during aging? And I've mentioned already that collagen is quite a long-lived molecule, so its half-life is like 10-15 years, dependent on the tissue and cartilage. In cartilage, it's even more than that. But when the molecule lives that long, it is subject to a very slow process that is called glycation. And glycation is basically a very, very natural process when you have sugar molecules, and we have plenty of sugar molecules in our body, obviously. And when you have like some temperature greater than 0, then the Maillard reaction happens. And what is Maillard reaction? It is when sugar attaches to one of the amino acid residues, and then two things can happen. One thing can happen is like this glucose molecule or some other sugar molecule stays attached to the collagen strand, attached with one end, and the other is free floating. And this is called adduct. And another thing, what happens is when the molecule kind of cross-links, kind of glues two collagen molecules together. And this is called, like, obviously cross-link. And we can say, okay, what's the problem with those? So these adducts, I'll start from them. They probably, again, I don't have like 100% bulletproof evidence, but cells have so-called receptor for advanced glycation end products. These are very, very ancient receptors of innate immunity, and they react with these glycation products and cause inflammation. And so my idea is basically that since more and more adducts accumulate in the extracellular matrix, they interact with rages on the surface of cell membranes of different cells, including stem cells, and basically trigger the inflammation. And this localized inflammation causes this, like, a lot of different things, including this clonal hematopoiesis that we discussed just recently. And this is one problem. And what can we do with that? We have a pretty good idea. So, for instance, a company called Rebel Pharmaceuticals, they have reported that they have created an antibody that targets CML, carboxymethyl lysine, which is one of these adducts. So theoretically, we can basically reverse some fraction of this adducts, some fraction of carboxymethyl lysines or some other adducts. So this is theoretically possible to, detoxify, so deactivate this inflammation triggers, right? But it will require a lot of work to create an antibodies, catalytic antibodies that can be delivered in therapeutic quantities into the body without causing harm. But that's feasible. That's technically pretty feasible.
Yeah, and I think that's a very important distinction, antibody versus catabody, because
Yes.
you're not talking about not everybody might know the
Yes.
difference there, but
Yes.
like an antibody would intermediate like an immune response,
Yes.
which is not actually what you would want. What you want to do
Exactly.
is actually just break that cross-link off.
Exactly. So this is adduct. And this is basically, this is a, like, kind of reversal. So this is a reversive therapy. This will potentially remove this damage like forever. So, and that is kind of like a very good thing. But this is not our only one option, because what we can do, we can, of course, try to mitigate this inflammation without removing these adducts, right? And by targeting very specifically these specific cytokines, pathways, we can do that, and it already works. So anti-inflammatory medicine and inflammatory drugs like canakinumab reduced mortality by 33% in a randomized controlled trial, which is already something. And then, so we have kind of understanding what we can do with this type of damage, right? We can try to develop a catalytic antibody that can clear these adducts. But meanwhile, we have an option to, at least mitigate the damage that these things create by, like, targeting inflammation. Then the second thing is this crosslink. This is a thing that makes collagen stiff, but also this is the thing that makes collagen insoluble. And this is very important thing because I haven't thought of it before, right? So what collagen insolubility has to do with aging? But in fact, it is an interesting thing. So first of all, we know that it is a fact that in this insoluble collagen, or the degree of solubility of the collagen in human skin, for instance, increases with time, right? And diabetes exacerbates the effects of old age. And we also know that glycation, in vitro leads to poor solubility of collagen. And it can be like just a very interesting fact, but nothing more, if we didn't know about this research, which showed that if we introduce a mutation in the collagen molecule, which renders this collagen more resistant to collagenize, then we basically get a premature aging. So this mutation basically mimics the effect of glycation on the collagen, and this what naturally occurs in our body with time. So here, all collagen is resistant, and that's why effects are extremely rapid. So mice die in like several months. And the extremely interesting fact that these mice, you can see how many senescent cells in their blood vessels. So basically, within a couple of months, they develop so many senescent cells that basically normal mice develop after 2 years of normal life, right? So senescent cells somehow are linked to collagen insolubility, and this is a matter of fact, and this is totally overlooked.
Yeah, although I think we have to be very careful about accelerated aging models because it's a lot easier to break things than it is to improve things. Like a demonstration of like taking like a shorter lived animal and giving it better collagen repair abilities that was transferred from a longer lived animal, I think would be a better proof of this case.
Of course, it's not a proof.
Of course, it's more expensive and it takes longer.
It's not a proof. Yeah. The proof would be like making a collagen soluble again, right? But that solves the problem, because to prove, we need to create a therapy that basically renders the collagen soluble again. And this problem, unfortunately,
you could do a germline engineered inducible system where you switch the organism from producing one type of collagen to another, perhaps. I don't know.
Yeah, but okay. So the main, I kind of foresee the questions and the challenges from other sides that say, okay, this is mutation in collagen. Maybe apart from making collagen resistant, this mutation does something else, right? So you never know. Of course, you never know. But you have lots of pieces of puzzle that do not say decisively that this is in favor of this hypothesis. But in my opinion, this is a substantial piece of evidence in favor, alongside with other pieces of evidence that we already have, connecting extracellular matrix and other hallmarks of aging and mechanisms of aging and so on and so forth. But when I saw it, I realized, okay, people may not believe me, but you will see. It is a thing. So, yeah. So how to explain the fact, how to explain the connection between this collagen insolubility and cellular senescence? And I discovered quite some things that were like surprising for me. So, because previously we thought that cellular senescence is sort of a anti-cancer program that basically cells cease to divide, basically to stop proliferate, to reduce risk of cancers and whatnot. But already from the experiments with senolytics, we know that senescent cell clearance reduces rate of cancer, right? Mice had fewer tumors than control group. And this led me to, thinking that, okay, maybe it's like cellular senescence is not about like cancer, maybe it's something else. And then there is a strong link to matrix. cellular senescence is in some papers, maybe some version of cellular senescence. Maybe there are several types of cellular senescence, but at least some variants of cellular senescence most likely represent an anti-fibrotic program, because numerous studies say that, okay, if we remove these senescent cells, fibrosis is exacerbated. If we induce cellular senescence, fibrosis is reduced, and so on and so forth. And then, basically my hypothesis emerged that I say, okay, this collagen insolubility during aging, progressive collagen insolubility mimics fibrosis, and cells erroneously trigger this anti-fibrosis program, which is cellular senescence, and that's why insolubility of collagen is not a one-time thing, because it progressively increases. So cellular senescence progressively kicks in and tries to counteract the pseudofibrosis, so to speak. And yeah, this hypothesis is of course not proven yet. So this would be nice if someone try to prove it in vitro or in vivo. But yeah, so this is the idea. And basically, I do not have an answer how we can make a collagen soluble again, because collagen insolubility is mediated by these cross-links that basically pull different threads of collagen together, and they prevent the collagenase to sit down on the collagen molecule and find the pattern which basically serves as an initial point of collagenase enzyme. And basically, so it renders it insoluble for collagenase, but most likely it will prevent any other enzyme to digest collagen, apart from very, very strong enzymes which are not specific at all. But that's also a bad thing, because non-specific enzymes will cut everything, and it's not a good thing. Small molecules may fit in, because glucose can fit in, then we can probably design other small molecules, but small molecules are also non-specific, right? So that's a problem. So maybe there are some things like sphere oligomers or other synthetic chemical compounds that are smaller than proteins, but a little bit larger than small molecules and a little bit more specific, can be designed. Maybe that's an answer to this problem, but to me it seems like an extremely difficult task to restore the collagen. Luckily, again, maybe we don't need to make collagen soluble again, right? Maybe we can just discover molecular mechanisms that trigger different responses from cells that make these cells non-functionable, or like making these cells functioning not properly, and basically counteracting those mechanisms, right? For instance, ROCK inhibitors or some other yet undiscovered pathways. This is also an option, so we don't need to fix it probably.
Yeah, although if you don't, but I mean there are physical problems though, right? Having the stiffening of the collagen
Yes.
is gonna, right? Like, so that sounds to me like more like a delay tactic than a long-term solution.
That's true. Like, but we can win time, right? Until chemistry becomes like more nanotechnology. Yeah. Anyway. So, but speaking of mechanical problems, so I don't think like stiff collagen is limiting that much. So yes, tendons become stiffer and maybe more prone to rupture, but I think we can live with that. It doesn't severely limit our lifespan, right? But what limits our lifespan is elastin. And this is something that we are currently working on, and pretty successfully, I have to say. So this is like very famous picture. Almost everyone who works on elastin knows this picture, and basically This shows the collagen from a young person on the left and from old person on the right. And this is basically what happens to collagen. As I mentioned, numerous studies show that it is very likely that collagen is deposited only in the very, very, very young age, so shortly after birth, and like 98% of collagen or something like that, I'm not sure about quantitative estimate, but anyway, is deposited at this period, and then it's the expression of the collagen elastin genes.
Are you talking about elastin, not collagen?
Yeah, elastin. Yeah, sorry.
Yeah. So okay.
Yeah, elastin genes that participate in elastogenesis. The expression basically plummets, and there is virtually no renewal of this elastin mesh, and it leads to inevitable degradation of elastin, even if we take out other factors like chemical damage, whatever. Just pure mechanical stress is enough to degrade the elastin network. And this is, of course, a problem because there are lots of other stuff apart from mechanical stress, mechanical fractures that are induced by mechanical stress, calcification, glycation, like peroxidation, and so on and so forth. Everything impacts the integrity of elastin in our blood vessels.
Can you give me, for the audience, a little bit of a description of like where elastin is in the body? Like, I mean, people will obviously think about like artery walls and things like that, but like, like this, what does this network look like? Where is it in the body? Is it everywhere, or is it?
Yeah, I think it's not everywhere, but it is probably in the most important pieces of our body, in the skin. First of all, I'm kidding, of course, but for many people, it's actually even more important than cardiovascular system or whatnot, because changes in our appearance with aging is mediated by a significant degree by this elastin degradation. So because it gives our skin the mechanical properties, the elasticity, the ability to counteract gravity, right? So basically, there are of course other aspects that make skin old, but elastin is, in my opinion, the majority of effects of aging on skin are mediated by elastin loss and degradation. The second thing is, of course, cardiovascular system. And this is extremely important because elasticity of the blood vessels, especially of the main blood vessel of aorta, is something that protects all our organs from the detrimental effect of blood flow that heart pumps with a huge force into microcapillaries.
Yeah, it's the same reason why when you have in your house you have like a pressure vessel upstream of the pump, so that you're not getting. And I think in your foresight video, you actually showed some very nice visuals of this, and we can refer people to that in the show notes as well.
And the thing is, another tissue where elastin plays a huge role is our lungs. And the role which elastin plays there is that when we inhale, we use muscles. But when we exhale, normally we don't do anything because of the elasticity. Our lungs expel the air just because of the returning back to normal state. But when you lose elastin, you have to apply force to exhale, and that is, of course, ruining the quality of life and reducing the efficacy of the respiratory system and whatnot. So, as I mentioned, the most detrimental effect on lifespan elastin has in cardiovascular system, in heart and major blood vessels. This is for sure. And interesting, there are a lot of causative studies. So, for instance, when scientists induced stiffness in blood vessels that supply blood to brain, like animals developed cognitive decline, which was like very fast and apparent. And I guess the same will happen with any other organ. So if it's kidney, then like inducing stiffness will obviously lead to worsening of the kidney function, and you can continue. Yeah, and there's a very nice article, a paper, scientific paper that I personally very like. Of course, it's also speculative, right? So, but here authors claim that okay, elastin is like limiting our maximum longevity because if we project the rate of loss of elastin, of elasticity of our arteries, it crosses zero somewhere around 115 years.
Lines up nicely with observations, actually,
Yeah.
of maximum lifespan.
Yeah.
So we could go into the biology a lot more, but I think you've made a pretty strong case for the importance of the ECM. And I think what we should probably talk about actually is what do we do about it? You've talked a little bit about the potential for drugs and catabodies, but I kind of want you to think big here. Like, what would be like the all-out, let's put together the Apollo program for solving ECM? What would that look like? Have you thought about that? Give us like, what are your ideas there?
So I think we should start from elastin. So because in my opinion, that's why I currently make a lot of efforts in this direction, because it will be a clear case, right? Elastin has a great potential to be the first real achievement in the field of anti-aging. So because, epigenetic rejuvenation sounds cool, but you cannot see it, right? But if you rejuvenate the elastin, you will see it immediately, and even if it doesn't increase your lifespan, people still will give much more credibility to anti-aging research in general. So, but if we would have like an unlimited amount of resources, of course we shouldn't focus on one thing. We should do multiple things in parallel, right? And as I mentioned, we first need to understand how exactly this aged ECM influence cells in vitro and vivo, right? Which molecular pathways and cascades are involved? How can we counteract this influence, right? And then experiment. So try to, mitigate the effect. And in parallel, simultaneously, we need to focus on developing new chemicals, try to understand, do molecular simulations, right? So these crosslinks target glucosapine, which is believed to be a major crosslink in the collagen. Maybe quantum, like computational quantum chemistry, will help us to do like a lot of studies in silico.
Yeah, I think that's an important distinction, is that like, so there's certain areas where computational biology is starting to allow us to replace wet lab work, which means that you can do the effort faster, more scalable, just add more GPUs.
Yeah.
So this would be an interesting use case there, where it's like, it's not as, people aspire to full cell simulation, but we're far from being able to do that. But if you could characterize all of the different things that are changing with the ECM, and then do some kind of exhaustive simulations of like, what are all the potential chemicals that could interact with those crosslinks?
Not just interact, do a specific thing, break it, because this is what we currently do not know how to do. Our in silico screening now boils down to just finding molecules that dock, like molecular docking, that they just bond with, or like not bond, but fit into some pocket of protein, right? And then luckily it interacts in a way that we would like to have. But here it is a completely different thing. So we need to just align the molecule properly, that it fits in there, but it also like forms some specific bonds, and as a result the crosslink breaks, right? And the more important thing, it doesn't recover again. So this should be like a terminal process, so that crosslink has like it breaks and it stops after that, reaches a terminal position.
What about other approaches too, though? Like, so in terms in modalities of thinking here, I mean, we do get remodeling of the ECM by macrophages.
Totally possible.
There's a lot, like, right. So,
Yes.
like, I know there's projects like the Chan Zuckerberg initiative has a new engineered cells project to try to create cells that are actually like able to do complicated repair functions. Do you think that's feasible to do that with elastin?
Yeah. Well, elastin or like collagen, like this cross-linked insoluble collagen, I think it's totally possible to try to maybe engineer or find an enzyme that can degrade this insoluble collagen, right? And then somehow in a controllable fashion, bit by bit, like solubilize parts of the collagen. So in my opinion, yeah, rejuvenation won't be a like very fast process. So it will be like a continuous process of multiple interventions. It won't be like a single drug that you take and you suddenly become young. So it's more like a tissue remodeling, continuous tissue remodeling. Yeah, so I think that's a possible, or maybe a combination of approaches. So we first like try to degrade like upper layers of collagen, and then maybe or like with small molecules we can make gaps between collagen strands a little bit wider, and then enzymes can go in, and yeah, so plenty of possibilities, but there needs to be like a lot of research in this topic. So I don't think it is infeasible. I think it's totally feasible, but we need to do in parallel a lot of things, but we need to accept the fact that it is likely that this thing plays a huge role in aging, right? But we need to accept the fact that yes, it might not be the like ultimate problem solver. So maybe, as you mentioned, if we fix it, it won't bring us like an extreme longevity, but we have to try, because without trying, we can't get an answer, right? We can argue about the importance of that, or argue about the programmable death theories and whatnot, but it won't bring us to reaching radical longevity.
So in the best case scenario, suppose we can, reverse or repair all these changes to the ECM. Do you think that could potentially get us all the way there to radical longevity? And maybe you can speculate on what radical longevity
Okay,
means.
yeah,
because I guess if I understand correctly through all of this, the main thesis is that through some observations and clues, you're saying that the cells can probably survive much longer on their own, right, outside the body, and you've demonstrated some of those experiments, or at least shared some of those experiments with us from the literature. And so if we just can just repair the microenvironment, then we could get some sort of a radical life extension. But do you think that's all the way, or do you think there'll still be some, I don't know, limiting factor? You said the DNA mutations aren't a big deal, but I'm just curious, like in the best case scenario, crack the ECM, repair it, what could we get?
Very, very good question. Very good question. So of course you never know, first of all, right? But my assumption, this is what I call strong ECM hypothesis, is that if we reverse all the changes in the ECM that lead to different problems, then cells without this negative influence from the outside, they restore their functionality. And even if there are clonal selection, as we discussed earlier, this clonal selection without this pressure from the old ECM, from the old environment, is clonal selection, it might turn the other way, and the healthy clones will take over. So it might be so that by repairing the ECM, we can actually bring whole organism to a younger state. And this is what I actually believe in. And yeah, I'm a supporter of strong ECM hypothesis.
So you're thinking of like potentially additional decades of life, not just like
Exactly. Not even decades. So
maybe more.
if we remove aging, what do you think is our expected lifespan?
Well, there's the extrinsic causes of death.
Yeah. If we only leave extrinsic causes of death at the rate that we currently have, without taking into account like future improvements.
Yeah. Well, the lowest mortality rate is in like 10 or 15-year-olds, I think. And those 1-year mortality rates for people that age, like 10 to 15, is like 1 in 10,000, something like that.
And without aging, the expected lifespan is 1 over that quantity. Yeah. So that's 10,000 years, right? For more realistic scenarios, like to take a 20-something-year-old man, and so this is like 1 in 1,000. So the mortality rate, right? So that gives us still 1,000 years of life expectancy. So this is radical longevity. We're not talking about like 10 decades of lifespan. We are talking about like hundreds of years of possible lifespan. Imagine what a human being can do during this time. Imagine how much of an experience, how much of a knowledge and a human being can gain during this time. And this actually enables at least partially, so not alone, in combination with other things, it enables interstellar travels, right? So that's crazy, yeah. But of course, like getting back to your question, of course, it might not be the case that reversing extracellular matrix aging will also like fully restore cellular functionality. But then we can help, right? We can help to restore by, modified cellular reprogramming in conjunction with ECM restoration. But I think that alone this won't work. So cellular reprogramming alone won't work without removing the constant pressure from the environment, from the aged ECM. But together they can work like amazingly well in theory. Then DNA damage. It's a very good point. So the fact that it doesn't seem to be causing like aging in normal conditions doesn't mean that it may limit longevity like at a very, very old age, right? But we will have a lot of time to discover that and find means to overcome this problem.
All right, so I think we've covered the ECM in a great amount of detail, and I think Nathan and I are looking forward to spending some in real life time with you to try to flesh out what the roadmap would actually look like to give it the best shot that we possibly could and the fastest progress we possibly could. But one of the things is we're trying to build a more comprehensive roadmap that looks at all of the different things that people are doing. And of course, you chose to work on this, which meant choosing not to work on other paths that other people are pursuing. And I think before we run out of time, it would be good to get your thoughts on some of the other paths that people are pursuing and perhaps why you didn't pursue them, whether you see any reasons why those paths would fail, or perhaps maybe you actually are optimistic and it's just there's only so much time in the day and you had to choose something for yourself. And so I think maybe, Nathan, you might want to start with asking some questions about replacement, because that's the one that's dear to your heart. Well, replacement and cryopreservation.
Yeah. Thanks for bringing that up, Mark. So one of the reasons we really wanted to talk to you for one of the first podcasts in this series is your paper that you co-wrote with Alexei Moskalev, the non-enzymatic stochastic changes in long-lived molecules. That's a paper that Jean Hebert, who's a professor at Albert Einstein College of Medicine, one of our friends, your friend as well, he cites that paper a lot for justifying his approach, actually. So he doesn't want to go down the path of, creating different molecules to try and break cross links or repair elastin or collagen. His idea is just like, why don't we just replace it completely? And so what do you think about that? Like, why not try that? Or do you think, I'm curious what you think are the, like, actually, I should also sort of like give a bit of background. So like Jean Hebert, basically what he's trying to do is progressively replace the neocortex with engineered tissue, iPSC-derived cells of brain cells, essentially, in the correct layers in a matrix, and progressively do that. And then other people in the replacement paradigm are working on, either organs or whole body replacement. People who are interested can look up Jean Hebert's work. But yeah, go ahead. Sorry.
I think, first of all, we, as I want to repeat myself, we don't have a luxury to stick to one approach, right? But to me, like full tissue replacement sounds like a more complicated task, actually. So to me, it's only, I perceive it as only looking easier, right? So just let's replace something from scratch rather than repair. Yeah, so if you have a car that has some damage in it, in a very, very rare cases, you will fully replace the car. So you probably will try to repair it until some critical point, right? Maybe for old person it makes sense, right, to replace. But again, so replace body, maybe. So maybe you can grow a clone in the future or something like that. But with brain, it is a bit problematic still. So because, you can add probably something to the brain, right? Some, maybe, as we saw in the experiment with mice, neural progenitor cells added to the rat brain. But the majority of brain is old, right? And the brain also has ECM. And the thing is that I think ECM in the brain can also be responsible for some cognitive problems. Here is another my hypothesis. So that the ECM that surrounds neurons, so-called like perineuronal net, you need to clear the extracellular matrix to form synapses, right? But remember what we discussed before. We discussed that matrix becomes, not necessarily the brain, but in general, becomes less soluble. This may happen to brain as well. Matrix in the brain may become less soluble, and it may hinder the ability to form new synapses. So what could happen is that we add new parts of the brain that are with good neuroplasticity, that are young, but majority of our information in the brain is stored in this old kind of insoluble brain that is very slow, and then it's unclear how everything will glue together. So I don't know, like maybe we can overcome these issues, but this still will require from us to understand.
Yeah, he, like with Jean Hebert, like one of the major outstanding questions is, if you grafted new tissue, sure, the new tissue will be new and young, but it needs to form connections with the other part of the brain. And if an aged ECM actually effectively blocks it from doing that, that could be something that has to be part of his strategy, is to be able to repair the ECM sufficiently that those long-range connections from the graft to the existing brain are capable of forming. Is this something you've talked with him about directly very much?
Yeah. We discussed it briefly, but not to, like, because we didn't have time to chat a lot face to face, right? So he lives in the US, I live in Europe, so it's kind of difficult to had a nice chat, a night-long chat, like like in science. But anyway, so one way to prove or disprove it is to try an old brain and graft new like organoid like in the old brain and see what happens, how well it integrates with the old brain. If it's okay, then it's okay. But if not, then Houston, we have a problem.
Yeah, definitely. And I think that's kind of like what John is doing first, like the proof of concepts in model organisms and obviously in non-human primates. And hopefully that works out, and we'll find out soon if that strategy is capable. Yeah. Okay, cool. So I think that covers the replacement part. And yeah, I agree. Like in terms of technical risks, that's always the big one, the brain, and taking the young tissue and seeing if it can reconnect to the old parts of the brain. Okay, and then so in our roadmap at OBF, we also, replacement is one of the paths that we identified as being plausible to getting us to radical life extension, maybe even indefinite lifespan extension. And it's an interesting one because it can sort of map out a reasonable time frame, let's say, and a budget. But some of the other approaches that we're thinking about also include, well, this one's not really solving aging. It's more just like buying time sort of situation, but like cryopreservation. So do you have any thoughts on this?
Yeah.
Well, thought about cryopreservation?
I kind of, in the field of transhumanism, because it's very connected. So of course, I studied lots of stuff in that domain, like from, I don't know, like cryopreservation to like, mind uploading and whatnot. In the very beginning, I was like super optimistic about cryopreservation of the brain and like of the entire body, but let's talk a bit about brain, right? And the major issue with cryopreservation is, ice crystals that we all know, and there is like a lot of counter arguments about, okay, there are cryoprotectants, whatnot, but let me talk a little bit for the audience. What is the problem like with naive cryopreservation when we just freeze the brains in like minus 200 degrees? Then the water that is in the cells, it forms the crystals, they expand and basically rupture cells, killing those cells. And basically, okay, we have cryoprotectors, right? So the chemical compounds that counteract this ability of water to form crystals, and basically we are saved. Okay, so we stop here, right? But it's actually not the case, because there is a prize for brain cryopreservation that basically evaluated 21 cm, or I don't remember what was exactly the organization that basically did it, but they evaluated the result of brain cryopreservation, whole brain cryopreservation after adding cryoprotectants. And though the brain looked very well intact from the macroscopic view, when they looked in the microscopic, like slices of the tissue, they discovered that, okay, basically all the synapses are not distinguishable under the electronic microscopy. So it kind of squished the brain, and basically also these cryoprotectants, they repel the water, but water is something that supports proteins, that prevents them from unfolding and aggregating with each other. So by using cryoprotectants, we basically introduce uncontrollable polymer, like aggregation of proteins. So in the best case, if we are able with some future technologies to rejuvenate, like to bring someone back, we will lose a lot of information that might be encoded in these proteins, right? And there is another argument that for myself, like conceptually, so why I'm not super huge fan of the cryopreservation. So if you think the whole idea about cryopreservation boils down to, emergence of some future tech that will restore you from I don't know what, right? So because we clearly know that after cryopreservation with cryoprotectants or without them, it's not possible to revive the tissue, the organism. We only may hope to restore some information, right, from the cryopreserved brain. But if we just wait for a couple of thousand or maybe tens of thousand years, maybe there will be a technology that will restore enough information from ash, right? Physics allows that. So if you know molecules, you can trace where they were, and so having enough computational power, it's possible to do. So, and while you are dead, you basically can wait whatever long you want. So I don't see conceptual benefit of that. So I would like not to die, not to experience the moment of death. But when I'm dead, for me, I don't care how many years. Right? And theoretically, yeah, you can take your brain, slice it, take a picture of it, and then store it as a digital, and hope that it will provide enough information to recover your identity. The same thing.
Yeah. So I guess there's some arguments, of course, against cryopreservation. I think people that we've talked to, right, who are very interested in cryopreservation and working on developing the technology and improving the process, would say things like, "Oh, well, compared to the alternatives, right? So maybe we don't have great confidence currently that we can revive people or anything like that from cryopreservation. But given the alternatives, like if you die now, your only alternatives are to be buried in the ground or to be turned into ashes, like you said. And I think there's probably a large difference between rearranging the atoms from ashes and the I guess the gases released from combustion,
exactly.
and back into versus something that's like cryopreserved. I think there's quite a big difference there in complexity.
Yeah, there are like alternatives to cryopreservation. You can like kind of fixate the brain in some chemical compound,
Plastinate,
which doesn't require cryogenic temperatures, so it is much more affordable to store the brain. So, but the effect is like we have to be honest with ourselves. So cryopreservation will most likely not allow to. It's not as some people think it's like I go to sleep. No, it's not. It's a different thing. So while during the cryopreservation, there are drastic changes in the molecular machinery of the brain. So it's not just you just freeze, molecules just stopped at places, and then when it backs to normal temperature, everything continues like it was before, and then electrical activity. No, there is likely no revival possible.
Well, so
I think so.
I would push back on that a little bit too. I mean, I think there are people that think reversible cryostasis is not infeasible, and
it's
part of the roadmap we're building will be what would it take to achieve that. And a lot of just as in like the ECM might be very important and complementary to Jean Hebert's replacement approaches, like my suspicion is that bioengineering technology, like genetic engineering technology, could potentially enable reversible cryostasis, because we actually already know that there are species that are frozen and thawed and resume their normal activity. And so we know that there's natural examples of it. And if we had sufficient engineering control of biology and genetic engineering methods, perhaps we could actually modify ourselves to be reversibly cryopreservable. the problem with that whole strategy is there's a lot of both science and technology problems of indeterminate complexity. We don't yet have the ability to make those genetic modifications or know what they would be. But a lot of people are bullish on that pathway, actually, that we could, build computational models of biology and improve gene delivery and gene editing to the point where we could do that level of modification of human biology.
Yeah, and I would also point out two other data points. So that cryopreservation, people would say as arguments, and I'm not an expert, but people can look up this stuff on their own. But yeah, like C. elegans, obviously a very small organism. Apparently, you can cryopreserve it, bring it back. The synapses are still intact. It can demonstrate some sort of behavior that is like a memory that can be preserved as well. So, I mean, I haven't looked deeply into that, but that's one of the arguments that cryopreservation people would make. And then the other one is more recent. There was a recent, I think, science paper where they demonstrated using like a nano-warming technique with cryoprotectant agents to cryopreserve a rat kidney, and then brought it down to cryogenic temperatures, and then revived it back, and then transplanted it back into a rat, and it was fully functional. Yeah, definitely not the same as a brain. Obviously, a lot smaller organ compared to, the brain of a human.
Wasn't it rabbit kidney?
No, this was a rat kidney, actually. The rabbit kidney stuff is, I think, more 21st century medicine, a long time ago. This nano-warming technique where they used iron oxide nanoparticles and a magnetic field, like an alternating magnetic field, to rapidly rewarm to prevent ice crystal formation on the rewarming side. This was more recent. This paper was just a few months ago in Science. Yeah.
Nice. Interesting. Yeah, but the scale, the size also matters, right? So you need to deliver these nanoparticles or whatever chemicals like uniformly across the organ. So you need to create enough pressure to do the perfusion, and it will also induce some damage to the organ. So it's a very complicated. I'm not saying this isn't feasible, so I will always refrain myself to say that it's something is impossible or something won't work, because science always brings surprises, and sometimes infeasible things become normal, previously infeasible things. But yeah, I think it's more challenging technologically to make it work, to make like fully reversible cryonics, in comparison to progressively tackle ECM and other problems with small steps, right? So first, like trying to slow down, mitigate slightly the influence of the old environment, then trying to reverse it, damage after damage. And yeah, so we have like a steady path. And each step on that path adds to our life expectancy. So we have more and more years to live with every progressive step on this roadmap.
Okay. And then there was one other part of our roadmap that we identified. Maybe Mark can talk about it. It's just bioengineering in general, and a lot of essentially full genetic control of the human body.
Yeah, I mean, a lot of people come to sort of a world of longevity and life extension from software backgrounds, and you can say that genetic code is just another code. The difference between like a human and a redwood tree or a bristlecone pine tree that lives 9,000 years old is differences in code. But the question is like, to make so it doesn't seem like it breaks any law of physics to probably modify a human to live much longer. The question is, what edits do you need to make, and how do you make them? And so like the big limiting factors there are the iteration time on our experiments is currently sort of way too slow to make much progress in our lifetimes. We would need to have computational models in order to speed that up. And we also would need to be able to make those genetic modifications to ourselves. And so we're going to be building out the roadmap on that side of it as well. And so I guess I'm curious if you have any thoughts or on that side of the roadmap? Do you think it's feasible at all? Do you think it's complementary with work that you're doing? Do you know of any promising projects or approaches in that area that you think we should be looking at?
Yeah, so this is a vast area actually. So I will probably divide it into two parts. Like first is like gene editing, right? And then basically how can we, modify the genome to be able to produce like more long-living organism or something like that. And then maybe more practical things like gene therapies in aging. Yeah, so this is a very difficult question. So very difficult question. Of course, genetic differences play like a huge role in this is unquestionable. So because we know it for sure, and we know some genes that can yield several fold difference in lifespan within the same in the same organ. Like one mutation in C. elegans can extend lifespan more than 10 times or almost 20 times in the P3K pathway. This is out of the question. But the problem is, in my opinion, again, that most of these genetic differences act during the maturation or development of the organism. So if you have an adult person or like adult animal, it's already too late to try to alter the genome, because these things, for instance, just in connection to the ECM, maybe the ECM that was deposited with some mutations, it becomes like less susceptible to glycation. Just a wild hypothesis, for instance, or something like that. So the genetic instructions, you can change it in some fraction of cells, but it has little impact on the longevity of the organism, because most of the work was already done during the development of the organisms. So, and I think the majority of longevity genes work the same way, this way. I mean, but there are genes that increase lifespan, and presumably we can utilize their power during adulthood. So for instance, Clotho or like FGF21 or some other genes, right, that are known to induce longevity in adult organisms.
Yeah, I think I would agree with what you're saying in terms of what has been studied so far. And I think that's an artifact of us looking at, invertebrates. Like gerontology historically has looked at these short-lived animals. And then of course for the ECM, as you say, it's laid down and it's, that's done once in some cases and not done again. And so you would have had to have that modification from the beginning. But there's also, if you look at like sort of longer lived species like elephants, I think it was recently showed that they have just like extra copies of certain tumor gatekeeper genes, right? And so that could be added later.
That's theoretically it can be, but well, I can imagine, of course, if you just remove all your blood stem cells and replace them with genetically modified cells which harbor like additional copies of P53 gene or whatnot. Yeah, that's theoretically possible. But what to do with again with brain cells, right? With other tissues in the body. So I think it can be done, but it is very limited what you can do with this approach. Of course, it can be done, but it's not how people perceive it. So it's not that you magically like transform an organism into something else. So you have very limited capabilities of introducing this genetic modifications. So first of all, the delivery problem, right? So how would you deliver genes to like all cells in the body?
Yeah, no, there's definitely major technical challenges, and we'll be bringing on other people who will sort of steelman the case for what is possible in our lifetimes in that regard. But yeah, definitely it's not an easy problem.
I think one other question I really wanted to pick your brain on, and for other people that are working on kind of like drugs, pharmaceuticals, or monoclonal antibodies, whatever, to target certain molecular targets for aging. What's your mental model for actually demonstrating that you're successful, right? So the idea here is when people talk about aging, there's two things. Like some people think about aging as like just the phenotype of aging, or maybe diseases of aging and delaying that. But for people who are in our community at LBF, who are really concerned about actual lifespan, like actually demonstrating lifespan extension, that's a trickier proposition, right? How do you actually demonstrate this, right? That the therapeutic, the intervention that you might develop that repairs ECM or elastin, how do we demonstrate that this actually extends lifespan and that you're successful once you actually create this thing?
Super nice question. So very, very interesting and important one. I've been thinking about this for years, for more than a decade, right? And I was a huge fan of the concept of biological age, right? That we have a single number that predicts, our true age, so to speak, and then by difference with chronological age, we can tell something. But in fact, I'm becoming very skeptical about this concept, and because it cannot distinguish correlation from causation. That's a very, very core problem of science, right? Dissecting correlation and causation. And the all studies that create this definition of biological age, they are done on cross-sectional data, and cross-sectional data is very, very poor when it comes to causative effects. So there are so many biases. For instance, let's imagine that on the horizontal axis there is age, and on the vertical axis is some normalized values of some biomarker. And at first glance, it may seem that some biomarker declines with age, right? Then we may think, okay, we may want to increase the value of this biomarker, and then we rejuvenate the person. But how this data was generated? This data was generated with a very simple procedure. So we have initial normal distribution of some value of biomarker, and then people with higher value of this biomarker have a higher chance to die. And it's basically less probability, and the people with higher values of the marker initially have less likelihood to live to 90, 100 years. And that's why we see this decline. So we capture exactly opposite from this cross-sectional data than it is in real life. So, and this bias is just only one single example of multiple biases there that are in the data. So that's why I don't really trust almost any measure of biological age, especially epigenetic clocks and what is associated with it. I believe in functional biomarkers more now.
We asked for a hot take, and I think this was a good one.
Yeah. I believe now more in functional biomarkers of aging. So if you show me a group, scientific group or whatever, that took a person, like 90-year-old person, and basically did some intervention, this person ran 100-meter dash under 15 seconds at 90 years old. So that I will buy this, because in my opinion, this will be undoubtedly this will be correlated with increased lifespan. And even if not, it already enormously increases the quality of life and health span. So this is already a huge achievement.
Yeah.
So I think we should aim for reversing age-related phenotypes and strictly measurable markers like VO2 max, hand grip strengths, I don't know, like time on some distance, like running some distance, elasticity of blood vessels. This is also an objective measure and interpretable measure of what's happening with our body. Also, some markers are good, like maybe like how many lymphoid progenitors in the blood, what is the percentage of this clonal hematopoiesis, like this meaningful measurements that we can reverse, right? That we have a biological meaning, while, for instance, composite biomarkers like epigenetic age, we have no clue what's behind them. It's a black box for us, and people now try to run clinical trials and brag about like reducing epigenetic age by X years. It doesn't tell you anything.
Yeah, I think people are attempting to connect the particular epigenetic sites to actual biology and more advanced versions of the clocks. Like that is progress that is being worked on. And another thing we can do is, like, for instance, you can look at a biomarker. Biomarkers often change even from like if you measure them at different times of day, right? Which there can be a lot of noise.
Yes.
But things like a glucose stress test, right? Like, so if you're not taking a single data point or even longitudinal data points, might be less interesting than looking how that biomarker reacts to a stress. Like, give somebody a lot of sugar, see how quickly they, like, that's the standard thing that we do in order. So maybe stress tests and like measuring the ability of the biological systems to restore equilibrium is probably another good way to measure age.
Yes. Also, like other objective biomarkers, for instance, the size of the atherosclerotic plaque, for instance, something like that. So something that happens with aging, we know that it leads to some unwanted changes, and if we removed it or reduced, I don't know, size or detrimental effects, so then it's good and it's objectively good. And there are plenty of those biomarkers.
So we've been going for a while now, and I think there's one last sort of section that we wanted to cover before we take too much of your time, and I would say the rate of progress, I think, is a very important thing to consider. Like, are we actually making progress on these things fast enough for it to be relevant to humans that are living today? And so a major category of our roadmap is sort of ways to accelerate progress / breaking bottlenecks to progress. And that can be either technological or institutional innovations, right? Because there's issues on rate of progress, both on the science and technology side and on the sort of organizational institutional side. And so maybe as a final segment, what are your personal limiting factors on the rate of progress you can make? And then maybe thinking about the wider community as well. Like what bottlenecks exist and what things could accelerate progress?
Yeah, very important question. So as per your first question, so what is the likelihood to achieve age reversal, or I don't know, like radical life extension? Is there a chance for people that live today to achieve this goal, this life extension? It depends heavily on the age of a person that lives today. I evaluate my chances as to be pretty high, because even if we fix the current rate of progress in life extension, so I have roughly 50 years left, plus some years that I get from the normal progress of the medical healthcare. So it's over 65 years, and there is a high chance that during these 65 years, some breakthroughs will happen, and I'll get even more years to live, right? For people who have remaining life expectancy 10, 20 years, it's not so bright, in my opinion. So maybe we won't be able to solve the problem by that time. And as per bottlenecks, I think the biggest bottleneck is that all the progress happens in a commercial big pharma way, right? So it is just commercially not viable to create an anti-aging startup, because the whole model of commercializing like biomedical startups is aimed mostly on rare genetic diseases like orphan diseases, because they give you FDA fast track, they give you like very, very long time when you have like a priority for your like patents, and you can get money from it. And so aging, it's not clear how to commercialize this. It's really not clear, and that's why investors are very reluctant to invest. So that's why most startups focus on some specific age-related disease or condition, and it's not enough because we need to look deeper in the mechanisms of aging, like underlying, and the underlying mechanism, they do not have necessarily a specific target, right? A specific like manifestation. It's like cause of multiple things. And to solve the problem, we need, of course, we need funding. So like it's a no-brainer, but we can't get these funds in a traditional way, while like pharma way of doing things, right? So because we cannot commercialize it properly. On the other hand, we can get funding for fundamental science, but aging, in my opinion, I will say like maybe controversial thing, but aging is nowadays less about science, but more about engineering. We know already a lot of stuff which we can fix by applying bioengineering methods, techniques, whatnot. Yes, it's not simple, but it is also not a black box for us anymore. We know quite a lot, and we need to focus. While when we secure funds for science, for scientists, they do science, they try to uncover something new, while we don't need to uncover something new, we need to solve actual problem. So we need grants for bioengineers and not scientists, because scientists do science publications, but it doesn't bring us personally to radical longevity.
You're nailing a really important point there, in that there is a community and a set of institutions built around science, and then we have the private industry that is good at commercializing things that are close to being ready. But I often call it technology development, but also bioengineering would be another way. There's a lack of focus on that by funders, both on the public and private side. And it's because I think our institutions were designed to solve 20th century health problems. They weren't designed to solve a problem like aging. And we actually need to change our institutions to have a more engineering-like mindset, both like the academic, the government, and the private institutions need a shift in mindset to an engineering mindset. I think you're correct. I do think that there's probably still a lot that we don't know about aging, so I wouldn't want to stop doing the science.
Definitely.
But there's also a lot that we do know. There's a lot that we do know, and
but
we're just not actually doing much about it.
Exactly. Yeah. If you look at the number of money that goes into research of caloric restriction, you will be very surprised in comparison to what goes into, I don't know, like gene therapies of aging. So that always makes me laugh.
Yeah, I mean, one of the things we've identified as we're putting our roadmap together is that there's a lot of critical path work that in its fully funded form would still be a rounding error on things that humans spend a lot more money on.
Exactly.
Right? which is a very unfortunate situation to be in.
We don't need extremely huge amount of money. So with wise spending, I think,
but we need a lot more than we have right now.
Exactly. So because we need because current funding is around zero, so to speak, on a logarithmic scale, we need magnitudes more. But it's I would say much less than the money that were spent on Apollo program or on the moonshot program. So in taking into account inflation, for instance, and this will already be enough to deliver some tangible results, and this will increase trust, and I think it will bring more money into the field. But without money spent on practical implementations of the current knowledge about aging, we can't move forward, because currently there is this vicious cycle. We don't have we cannot demonstrate anything because we have no money, and we can't get no money because the field has no credibility because we haven't delivered anything yet. We have to bring this vicious cycle by delivering something that will
All right. Well, I hope people out there listening to this want to get involved and help break these bottlenecks.
Yeah, we can make short clips and from the most outrageous claims.
Yeah. Okay, great. So I think we've taken up a lot of your time. It's been we've been going for almost.
No. It's a pleasure to chat, and topic is very interesting.
Yeah.
And, so no problem. It's
it was great chatting with you, Alex. And where can people find your work or connect with you? What's the best way?
That's a good question. So I think we have an email, our Radical Life Extension group. We have an email, so we can, if it's possible, we can link it in the description somewhere. And so well,
definitely. Yeah, we can put it in the show notes. And are you on LinkedIn, Twitter?
I'm on LinkedIn. I'm on Facebook. But yeah, so.
And aside from money, which obviously is something people are always looking for, is there any other type of specific support that could help your work go faster? Are you looking for any particular types of talent, people, things like that?
Yeah, I think it's definitely not only money, but also relevant connection in the field. So first of all, we already kind of, I think one thing that it can be already done right now is that testing actual interventions on humans, right? So because mice is a good model, but it's by no means it is a human being, right? And we already have something that works, and we already have some things that I mentioned that we can use as a good indicators of the efficiency of the therapy, right? And creating a network of biohackers, doctors, medical doctors. And I heard there is now there is a good movement towards the legislation. So am I right that in some certain places in the US, for instance, you now are able to test drugs or therapies on yourself, right? Is it correct?
It's the right to try law was passed in Montana, I think, with the help of A4LI, which is an organization that, if anyone is listening, also could use support in getting more of that type of legislation passed, because it's basically just one guy pushing this right now, and he's actually had quite a bit of success with a very small amount of money.
Yeah, I think this is extremely important, because there is something that can be already tried. And so this is a little bit of a teaser. We are not sure yet about the results, but anyway, so we know at least 2 cases of a significant reduction of the atherosclerotic plaque. So by significant, I mean like by like 30% or something, while typically in clinical trial there is like several percent reduction is already considered to be significant. So even with current medicine, current drugs, therapies, it is already achievable, to do better than current best official medicine can do.
Well, that sounds like a really good note to end on. Unless anyone else has any final comments, I just want to thank you so much for your time today, Alexander, and we'll do our best to get this information to as wide an audience as we can.
Thank you very much for inviting me. It was a pleasure. And I hope, this will be like a first step, a very good first step in defeating aging and achieving radical longevity. So, thanks again.
Awesome. Yeah, thanks, Alex.